What's up, biology students? Mr. Holloway here. Today's video is a bit of a history lesson about some of the key scientific discoveries that helped us to understand the basic structure and function of DNA. Many of these discoveries have only occurred within the past 60 to 70 years or so, and new discoveries are still being made all the time. Although we're learning more about genetics and how it influences our lives every day, we already know quite a bit about the molecule we call DNA. For example, we know that DNA is a polymer composed of many monomers called nucleotides. We know that each of these nucleotides is composed of a 5-carbon sugar, a phosphate group, and one of several nitrogenous bases. We know that these nucleotides are organized into 2 anti-parallel strands, which means that these strands run in opposite directions. These anti-parallel strands are also plectonemic, meaning they wrap around each other in a double helix. We know that the 2 strands are held together by hydrogen bonds that form between nitrogenous bases, or base pairs. We also know that there are certain rules that dictate which nitrogenous bases can form hydrogen bonds. A always bonds to T, and G always bonds to C. Furthermore, we know that the order, or sequence of these nitrogenous bases determines the kind of proteins that a cell is able to manufacture, and that the function of these proteins ultimately gives an organism its traits. And when I say we know these things, I mean that we suspect these things to be true based on a tremendous amount of scientific data and observations collected over the past 60 to 70 years. We didn't always know these things, and we certainly didn't learn them overnight. So if you are ever feeling as though you are having a difficult time remembering and understanding the stuff we talk about in this video, remember, it took thousands of scientists and over 70 years for us to generate this knowledge. The history of scientific discoveries that have helped us to generate this knowledge is, in fact, the topic of today's videos. That said, we're really only scratching the surface, and we'll only be focusing on a few of the many key discoveries that help us to better understand the structure and function of this really important biological molecule. Our story today begins in the 1920s, and features a British scientist named Frederick Griffith. Griffith was studying bacteria, specifically the bacteria that caused pneumonia. What gave these bacteria the ability to make us sick, while other kinds of bacteria are harmless? Griffith wanted to know, so he conducted some investigations. In one experiment, Griffith injected several mice with either the smooth, or S strain of bacteria, or a rough, or R strain of bacteria. The mice injected with the S strain died of pneumonia, and the mice injected with the R strain did not. In a follow-up experiment, Griffith used heat to kill the disease-causing S-strain bacteria, because heat causes the bacterial cells to denature and burst open, releasing their contents. He injected these heat-killed bacteria and their contents into more mice, and these mice lived. Killing the disease-causing S-strain bacteria prevented the disease from killing the mice. After that, Griffith took a mixture of heat-killed, disease-causing S-strain bacteria and live, harmless R-strain bacteria, and injected this mixture into yet more mice. And interestingly, these mice died of pneumonia as well, and an autopsy revealed live S-strain bacteria living in the lungs of the infected mice. The R-strain bacteria did not kill the mice before, and neither did the heat-killed, disease-causing S-strain bacteria. So why did the mixture of these two cause the mice to die? And why were there live S-strain bacteria growing inside of these mice when the S-strain bacteria added to the mice were clearly dead? Griffith reasoned that some chemical factor inside the bacteria was capable of changing harmless bacteria into disease-causing bacteria, and that this chemical must have been transferred from the heat-killed cells of the S strain into the live cells of the R strain. He called this process transformation because the harmless bacteria had literally been transformed into a different kind of bacteria, this time a harmful one. Griffith's experiment was groundbreaking at the time, and was an important step because it revealed to us that something inside the cell makes an organism what it is. More than that, it revealed that whatever this chemical factor was, it could be recognized by other cells, and had the ability to influence the function of other cells as well. Griffith didn't know what this chemical factor was, but knowing that the function of a cell is determined by something inside the cell, and not by some external factor, allows us to ask additional questions about what this mysterious transforming chemical factor is, and helps us to design new experiments to increase our knowledge. And that brings us to another scientific discovery, made by one Oswald Avery, a Canadian-born scientist working in the United States. In 1944, with World War II raging on outside, Oswald Avery was following in Frederick Griffith's footsteps, trying to determine what, exactly, was inside a bacterial cell that could cause transformation. At this point, we knew that cells contain several different kinds of important biological molecules: lipids, carbohydrates, proteins, and nucleic acids. But which of these molecules was responsible for transformation? Avery repeated Griffith's experiment, except he added an extra step. After using heat to kill the harmful S-strain bacteria, Avery systematically isolated each kind of molecule and exposed the harmless R-strain bacteria to each variety individually. In one trial, he kept only the lipids from the harmful bacteria and exposed the harmless bacteria to these. In another trial, he kept only the carbohydrates or kept only the proteins, but in each case, transformation did not occur. Only when Avery kept the nucleic acids and discarded the lipids, carbohydrates, and proteins did transformation still occur. Avery even went so far as to separate the nucleic acids into DNA and RNA, but only DNA was able to cause the harmless bacteria to transform into the disease-causing strain. These results suggested to Avery that out of all the molecules in the cell, all the lipids, proteins, carbohydrates, and nucleic acids, only DNA had the power to transform a bacterium from one kind into another. DNA was the chemical factor that Griffith had been referring to, and only DNA seemed to have the capacity to influence the fundamental nature of a cell. Today we know that bacteria are somewhat unique in their ability to absorb and use DNA from their environment. Fish don't absorb whale DNA and become whales, for example, and humans can't absorb DNA from a carrot that we've eaten in order to become a carrot, but our cells are able to take the nucleotides from carrot DNA and rearrange them to match our own DNA, which is how our cells are able to synthesize new DNA molecules when preparing for cell division. But there are other ways to transfer DNA from one organism to another in a usable way. Viruses do this all the time, in fact. In the early 1950s, Alfred Hershey and Martha Chase were conducting research on viruses. Specifically, they were studying a kind of virus called a bacteriophage, which literally means bacteria eater. Bacteriophages are viruses that attack and infect bacteria, and bacteriophages use bacterial cells in order to reproduce themselves. Viruses are weird, to say the least. They are not made of cells, they do not really grow or develop over time, and they do not consume any nutrients. They are able to reproduce, but only by hijacking the machinery of a living cell, and viruses have evolved over time to take on new forms and functions. Although they are not technically considered to be alive, viruses do contain genetic material, either DNA or RNA. This genetic material is encased in a protein shell called a capsid. Basically, a virus is a set of genes surrounded by a protein shell. In order to reproduce, viruses infect living cells by injecting their genetic material into that cell. The cell recognizes this genetic material, and because all DNA and RNA is fundamentally similar, the cell is able to read and interpret this genetic material as well. The viral genes injected into a cell contain instructions for assembling new viruses. So, since the cell doesn't know any better, it reads and acts upon these instructions, making many new copies of the virus that eventually burst out of the infected cell and can now find and infect new cells. This process happens in one of two ways. Either the cell acts upon these genes right away before dividing, which is depicted here as the lytic cycle, or the viral genes lay dormant for a while, allowing the cell to reproduce and pass on copies of these viral genes to many new cells. Eventually, this means that many new cells will contain instructions for making new viruses. And when these genes are finally turned on, that means that many cells will be making new viruses all at the same time. This second pattern is depicted here as the lysogenic cycle, and which pathway a cell takes depends on what kind of virus we're talking about. We know all this today, but back in the 1950s, we were only just starting to learn this information. At the time Hershey and Chase were conducting their research, we knew that viruses injected something into a cell, but we didn't know exactly what that something was. Whatever it was, it seemed to have the ability to reprogram the infected cell, tricking this cell into making new copies of the virus. But what was it that the viruses were injecting into the cells they infected? What could cause these cells to make new viruses when doing so ultimately caused the destruction of the cell? Hershey and Chase decided to find out. Hershey and Chase grew viruses in the lab, in cultures containing either radioactive phosphorus or radioactive sulfur. Because DNA contains phosphorus but no sulfur, and because proteins contain sulfur but no phosphorus, the radioactive phosphorus would only be found in a virus's DNA, and radioactive sulfur would only be found in the virus's proteins. Hershey and Chase were hoping that they could determine which of these two molecules were being transferred from the virus to its host by looking for radioactivity inside the host cell following infection by the virus. When the viruses grown in the culture containing radioactive phosphorus were allowed to infect bacterial cells, Hershey and Chase detected radioactive material inside the bacterial cells afterward. But when the viruses grown in the culture containing radioactive sulfur were allowed to infect bacterial cells, no such radiation was detected in the bacteria following infection. Since only radioactive phosphorus was transferred from the virus to the bacteria, but not radioactive sulfur, Hershey and Chase concluded that it was the DNA and not the proteins that a virus used to reprogram the host cell. This DNA was providing the instructions necessary for the cell to make more viruses. And since we know that humans and many other organisms are susceptible to infection by viruses, we can assume that the same thing applies to us. When we contract a virus, that virus is injecting its genetic material into our cells, and our cells are using this genetic material to make additional copies of the virus. Based on the work of Griffith, Avery, Hershey and Chase, as well as the work of many other scientists that we don't have time to talk about here, we were able to determine the function of DNA. We discovered that DNA contained information that instructs the cell in what to do and what kind of molecules to make. We also discovered that these instructions can be read by other organisms, and therefore must be made of fundamentally similar stuff. As we were learning more about the function of DNA, we were also learning a lot about the structure of DNA. Chemical analysis could tell us what DNA was made of, but how were those pieces put together? Erwin Chargaff, an Austrian scientist who came to the USA to escape the Nazi regime, helped us to discover a key piece of this puzzle. By analyzing the chemical composition of DNA from many different species, Chargaff discovered that the composition of DNA varied from species to species, in particular in regards to the nitrogenous bases. The relative amount of sugars and phosphates was pretty much the same from one species to the next, but species varied widely in terms of the relative amount of G's, A's, T's, and C's. Could it be these bases that distinguish one organism from another? Moreover, Chargaff discovered that in all organisms studied, the amount of A, or adenine bases, was always equal to the amount of T, or thymine bases. Likewise, he found that the amount of G, or guanine bases, is always equal to the amount of C, or cytosine bases. We call this Chargaff's rule. A always equals T, and G always equals C, in terms of its abundance within a particular DNA molecule from a particular species. But what this means, Chargaff couldn't say. Future scientists would see Chargaff's rule as indication of base pairing, but that part of the story comes later. Around the same time, Rosalind Franklin, a British scientist, was attempting to use X-rays to study the structure of DNA. After a great deal of experimentation and refining, she was able to produce this famous image. This X in the image suggests that DNA is a double-stranded molecule, and that these strands are coiled around one another. Here's where things get a little weird. This photo was one of the biggest clues revealing the structure of DNA, but in an era where women often didn't receive the respect they deserved, Rosalind Franklin's specific contributions went underappreciated for quite some time. She was accused of not knowing how to interpret her data, and even when others used her data and photos as the basis for their models of DNA, Franklin received very little credit for her contributions. A male colleague of Franklin's even gave away her photos to a competing pair of scientists without her permission, allowing these scientists to obtain a key piece of information they needed to build a complete model of DNA. These scientists were James Watson and Francis Crick, who did eventually come around to thank and acknowledge Franklin's work and contributions to the field, but not before publishing their work and claiming credit for constructing the first three-dimensional model of a DNA molecule. In fact, they were the first to build such a model, but they would likely not have been able to do so without Rosalind Franklin's photos. Interestingly, although Watson and Crick are credited with discovering the structure of DNA, they did so without conducting a single experiment. They built their model, which we can see here in this photo, based on the published and sometimes unpublished work of countless other scientists, most notably Chargaff and Franklin. It was Watson and Crick who proposed that the rules of base pairing could explain Chargaff's rule. The fact that A always bonds to T and the fact that G always bonds to C could explain why we find the same amount of adenine and thymine and the same amount of guanine and cytosine. Base pairing could also explain how the two strands of DNA were held together. They used Rosalind Franklin's photo to determine that DNA not only was double-stranded, but was coiled in a double helix, as we can see in their model, and in this rotating 3D model that we've seen several times now during this unit. The model built by Watson and Crick is still the standard used today, and although they conducted no experiments of their own, the fact that they were able to put together all of the pieces from all of the different experiments from all of the different scientists was a huge breakthrough. Today we accept that structure influences function at all levels of biology, and much of what we have learned about DNA since Watson and Crick is based on this structural model they built back in 1953. Our knowledge of DNA and its influence on our existence as living organisms is still growing, so the story is far from over. As this knowledge develops, it may help us to devise new medicines tailored specifically for the individual based on their genes, and is already helping us to engineer new organisms capable of meeting our needs. For example, the insulin shots taken by diabetics are produced by E. coli bacteria who have had the gene for human insulin spliced into their genetic code. These bacteria have been genetically engineered to produce medicine for human needs, and the possibilities do not end there. And with that, I will bring our video to a close. Thanks for watching, and remember, you can go back and watch this video as many times as you need until you feel like you understand how the scientists in this video have contributed to our knowledge of the structure and function of DNA.